Before we get started, I'd like to give a quick introduction of our panel members, who are all members of the UCI Center for Virus Research. First up is Michael Bookmeyer. He's a professor in the Division of Infectious Disease, Department of Medicine, and the UCI School of Medicine. He's also in the Department of Molecular Biology and Biochemistry in the School of Biological Sciences, and he's the Associate Director for the UCI Center for Virus Research. Mike will tell us about coronavirus biology essentials, genome structure and function, cell entry, replication, virus production. Next up will be Professor Don Forthall. He's the chief of our Division of Infectious Diseases in the Department of Medicine at the UCI School of Medicine. And Don is going to tell us about SARS-CoV-2 pathogenesis, translation from clinic to laboratory, regulatory and safety considerations, hosting the virus in model animals and tissue culture systems. Third up will be Ilham Massaoudi. She's a professor in the Department of Molecular Biology and Biochemistry in the UCI School of Biological Sciences. And Ilham will tell us about monitoring SARS interactions with the immune system, tools and caveats, and Ilham will also act as our moderator during this discussion. Finally, Professor Andrew Neumer from Department of Population Health and Disease Prevention, the UCI program in Public Health. Andrew will tell us about COVID-19 at the population level, studying spread and estimating mortality burden. Before handing this over to Ilham, I want to acknowledge the hard work of Professor Suzanne Sandmeyer, our Vice Dean of Research in the School of Medicine, and the staff in the School of Medicine Dean's Office for putting this event together on very short notice. It's greatly appreciated by all of us. And if we have time at the end of all 4 presentations, we will take questions for the speakers. And to do this, we will use the chat function on Zoom, and I'll try to monitor that for Ilham, and so you can submit the questions that way. So, Mike, without further ado, please.
Oh, I just got on it. Oh my God. Yeah, I just.
Unmute. Okay. All right. And can you see the screen now? My screen.
The answer is yes.
Okay, good. All right. So there we are. So the coronaviruses, the coronaviridae, are a family of positive stranded RNA viruses. They're non-segmented, and they largely infect vertebrates, although there have been some suggestions that they may infect some non-vertebrates as well. Those are not substantiated. This includes zoonotic and human infections of high pathogenicity. For example, SARS, COVID-19, which is the topic for today, infectious bronchitis virus of chickens, mouse hepatitis virus, and transmissible gastroenteritis virus of mice and pigs. They display a unique nested set messenger RNA structure, and because of that, the entire group has been named the nidovirales, and apparently nido means nested in Latin, although I never studied Latin. Now, how do I get forward now? All right, I am trying to move forward on this screen. There we go. Back one. There we go. All right. I want to straighten out one bit of confusion first before we go on about the terminology that's used to describe this virus. It's being called COVID-19 because it at the beginning of the research on it, it was identified as Wuhan virus, which is politically incorrect and inappropriate from a taxonomic point of view. The Committee on International Committee on Taxonomy of Viruses met quickly and discussed this and considered the data and considered the naming and the phylogeny of the virus. And the official name for it is actually SARS-CoV-2. So it really is a representative of the SARS family of viruses. The name 2019 nCoV was one of the original names, and COVID-19 really describes the disease, not the specifics. And that's indicated in this article, which is published in Nature Microbiology. I won't go into any more detail on that. The other aspect of this that's really important to understand is that this virus is not a totally unique virus. It belongs to a closely related family of viruses here at the top, which includes SARS-CoV coronavirus from 2003, and a number of animal isolates from bats, from rats, and from civet cats and other animals of coronaviruses in the SARS family. These are group 2 beta coronaviruses, and they share that relationship with MERS virus, which is a virus from the Middle East, and you can see it's more distantly related to SARS, and with human coronavirus OC43 here, and human coronaviruses HKU, Hong Kong University 1 and 24, which are also upper respiratory viruses. And then finally, NL63 and 229E belong to a different family of coronaviruses. They're basically distantly related. So that this group is actually a rather large group of phylogenetically related viruses. They exist mostly in bats. They're actually, the reservoir in nature is bats, and because bats have a defective innate immune response, they tolerate the virus. So they can be persistent in bats like many other animals, species are. In addition to the relationship, I want to show you the relationship in very schematic form here between SARS-CoV, SARS-CoV-2, which is the new virus, and MERS coronavirus. And what we've done here is we've illustrated the entire genome. And the color coding is simple. The darker the more related they are. And you can see 95 to 100% is here. And you can see that this is, first of all, SARS coronavirus on the top, SARS-CoV-2, and I just lost it again. Come on. Come on. There we go. Okay. SARS-CoV-2 has a number of genes that are very highly conserved, and it has others that are more distantly related. One point that I want to make, and really related to what Bert said a minute ago, is that the spike protein is critically important in the pathogenesis of this virus. And note that the N-terminal half of the spike protein is highly conserved between these two viruses. The C-terminal half, or what we call the S1 portion of the spike, is less conserved, although it is conserved to some degree, 80% here. And so those two differences exist. There are also some accessory genes that are slightly different, and structural genes that have some dissimilarity. We don't yet know what the full effect of those similarities and differences are in the virus. We do know that this is a very, very rapidly spreading virus. It's a virus of very high pathogenicity, and those are important points to keep in mind as we study that. MERS coronavirus, on the other hand, is very distantly related to all of these, although it has the same exact genome structure. Now, you heard also something about the size of this genome. Well, this genome is 31 to 32 kilobases long. To put that in perspective, poliovirus, which is the virus that Dr. Semler works with, is about 7 kb, so that if you went from genes 1 to 16 of this one, that would include 3 poliovirus genomes. So altogether, it's a little over 4 poliovirus genomes long. And the proteins look something like this. There's a leader sequence. There's a P65, which is one of the genes that helps take over the host, and then a multi-domain protein, NSP3, which actually has 15 active protein domains in it. So even a single domain like that is multifunctional. And then as you get up here toward this initial junction, this is actually a frame shift. It's not a stop codon, it's a frame shift. And then you get into the enzymes here that are involved in replication and editing of the genome. And then we get into the structural proteins, the spike protein, the envelope protein, the M protein, and the N protein at the far ends. Everything else here is either very minor structural proteins or immune modifiers or other molecules that are involved in replication. To give you an idea of where we stand with our knowledge of SARS, we published quite a lot on SARS back during the 2000s. And this is, for example, the model that we built of the replicase complex. NSP3 is this large domain here, and NSP3, NSP15, NSP16, 14, 13, 12, 7, and 8, and many copies of each are involved in making the replicase complex. So we did the nearest neighbor analysis, we did the mud pit, and that's what we found. The structure of the virus, idealized, looks something like this. It has a helical nucleocapsid that winds around the virus. It has spikes that penetrate the membrane but don't actually interact with the nucleocapsid. The M protein interacts with the nucleocapsid on the cytoplasmic side or on the intraviral side. So it's a fairly simple structure, a very robust structure, but it's enveloped, and the envelope derives from the host cytoplasmic membrane vesicles, not from the surface. SARS was identified as the agent of an outbreak of atypical pneumonia originating in Guangdong province in November of 2002. Like the case with this virus, we didn't find out about it except through word of mouth for about 2 months. It was kind of kept secret. But in spite of that, and this is in 2002, 2003, once the virus became known and the severity of the virus was known, at that time, a record was set for sequencing and full annotation of the genome in 30 days by 2 different laboratories. So the idea that we could begin to apply modern technology was first really hatched with SARS virus. The virus spread initially to, or eventually to 26 countries, resulting in about 9,000 cases, 8,098 cases, which looks like a very small number now compared to what we're dealing with, and 774 deaths. But one critical aspect of this is that it had a higher mortality rate per infected individual than the current SARS has. The host of this virus is the horseshoe bat, the Asian horseshoe bat. An intermediate host was the palm civet, who was traded on the wild animal markets and eaten. And the palm civet supported the virus replication without a lot of symptoms, so it was sold and eaten. And eventually spread to humans. And much like we're seeing now, SARS really shocked the world. The initial reports were that it was causing an atypical pneumonia, perhaps a pneumonitis. And these are some of the original images that came from CDC. Sharif Zaki at CDC did the initial analysis, and what Dr. Zaki showed was that, it was that you could see, first of all, multinucleate giant cells in the lungs, deep in the lungs. And here's the blow up of that one. And these multinucleate giant cells stained for the antigen of the virus. And then here, even in sputum, you can see viral particles in sputum at low power and high power here. So the virus was rather easy to detect, and because of the morphology of the virus and the nature of these multinucleate giant cells, and cross-reaction with other known coronaviruses, it was considered... Somebody talking to me? I'm sorry, I'm hearing you. So we undertook a number of different kinds of studies, and the first that we did was really a CryoEM study of the virus. And this was the first CryoEM study that was published in 2006 in Journal of Virology. The technique seems quaint now, but this was some of the original images that we got and the model that we built, which looks very similar actually to the previous model. This is the view of the spikes and the underlying structure of the M protein and the nucleocapsid. And this is looking top down on the spikes, which reveals that the spikes are trimeric. If you look at a deeper level, you find that the membrane protein is a lattice. This is just simply looking at the Fourier transform of that. So you can see here the membrane protein crossing like railroad ties on this bilayer. And that basically was the conclusion of the initial studies. The virus over the course of spread in southern China seemed to exist in different forms. And we're seeing really different pathogenicities of the new SARS virus now. And I wonder at this point whether we're not seeing a similar mutation process. It hasn't been documented, but it may well be. So it's something to look for. But what happened was that the virus mutated extensively in the spike protein. And this is in the receptor binding domain, which binds to the ACE2 receptor. And what you saw was the virus can be segregated into 3 groups, into a low pathogenicity group isolated in civets in 2004, Invasion of humans in 2003 and 2004 was represented by this sequence here. And then finally, high pathogenicity group and an epidemic group here in 2003 was found. So civets in 2003 and epidemic group in patients in 2003. So that you saw actually extensive mutations within a very limited, about 20 amino acid segment of the receptor binding domain of the spike protein. So again, among the things to watch for is changes in that region. And these are point mutations. If we look at the relationship with other viruses in the group, we have to remember that this is a member of a group that has a lot of similar viruses. Here's OC43, one of the human common cold viruses, bovine coronavirus, equine coronavirus, rat coronavirus. These are species that we've known about for a long time. And then finally, SARS virus is in here somewhere. The point is that this is a group of viruses, and they exist in group 1, group 2, and group 3 viruses. So this is a group 2b virus. So the final point that I want to make is that we know that bats are the natural reservoirs of SARS-like viruses. This is well established really in the course of the SARS epidemic. Since then, people have been studying these viruses for some time and have been surveying them in bats. And this is an example of one of these surveillance papers published in Nature Medicine in 2015. Ralph Barrick, who we know well, was the senior author. And the title of this paper, A SARS-like Cluster of Circulating Bat Coronaviruses Shows the Potential for Human Emergence. And there were half a dozen papers like this coming from Hong Kong, coming from Barrick's lab in North Carolina, coming from mainland China. And we should really have been ready and expecting something like this to happen. It shouldn't have surprised us, but in fact it has. And so that's the point that I would make in terms of really the public health surveillance and where they let us down. Finally, there's an emerging paradigm that this group has put together, an emergence paradigm, looking at how this virus might have evolved in hosts. And one is if you look at random mutations, a host range mutation. If you have a secondary host, you may get a human infection directly, or this may become an epidemic strain, and in the course of an epidemic, spread from a secondary animal like the palm civet and spread to a direct human infection or a secondary host reservoir. Once it's in the human, it can spread from human to human very rapidly. Another possibility is that you have a series of recombinational events that go on. This virus can recombine as well. And you can have a direct human infection directly from the bat. And again, a secondary host reservoir and an epidemic strain can evolve from that. So again, the secondary reservoir may in fact be a reservoir that we need to be concerned with, even if the problem is solved in humans. So I'm going to stop there and let's move on. Do you want me to answer questions at this point?
Mike, we'll save until the end.
Yes, thank you, Bert. Don, you're up.
Dr. Forthall.
I saw his face there.
Don, you need to unmute yourself and share your screen, please.
Sorry about that. Sorry, I'm trying to share my screen here. Tell me when you can see it.
Okay, I still can't see it.
Hmm.
Green button at the bottom of the bar.
Yes, that's the one I pressed.
Sometimes it takes a while to boot up.
Is your video activated?
Video is activated. When I
You're
share
on.
screen, I see something else. Anybody seeing my screen yet?
We see you.
How's that?
Perfect.
Got it.
Okay, all right. Sorry about that delay. Okay, I'm Don Forthall. Good afternoon, everybody. With my time, I thought I would first present a very brief clinical overview of COVID-19, being the clinician of the panel, and then pose some questions that I think are of interest to both clinicians and researchers, and that lend themselves to basic and translational research studies. So COVID-19 is mostly a fairly mild infection. About 80% are mild, and some of these, perhaps as many as 15 or 20%, are even completely asymptomatic. Patients with mild infection tend to have fever, myalgia, cough, and we're increasingly now seeing GI symptoms associated with these infections. But 15% will go on and have severe infection manifested by pneumonia and hypoxemia. 5% turn out critical, particularly with the development of the acute respiratory distress syndrome, ARDS, and also failure of other organs, particularly the kidney. And then we don't exactly know the case fatality rate yet, but it seems to be on the order of 1 to 2%. So clearly the upper and lower respiratory tract are the targets of this infection. Increasingly, it's looking like the GI tract is also infected by SARS-CoV-2. There's evidence now of not just PCR positive results from the stool, but also actual evidence of infectious virus in the stool. This shows a couple of images of the chest, CT scans. On the upper left is a rather mild infection showing what we call ground glass opacities in the lungs. This would be a mild to moderate case likely. The case on the right is a much more severe case. And in fact, this is consistent with an acute respiratory distress syndrome. It's important probably to mention what ARDS is, because you'll hear a lot about this when people talk about the management of cases and mortality due to COVID-19. And first of all, it's something we very commonly see in the ICU. In fact, if you walk through an ICU a year ago before we had COVID, probably 20% of the patients would be ventilated and have ARDS from various etiologies. And it's really a diagnosis that's almost always made clinically. There are certain histopathological features of it, but mostly it's a diagnosis made clinically, and it's characterized by new or worsening respiratory symptoms after a known insult, in this case an infection. It's also characterized by bilateral opacities on imaging, and then respiratory failure that's not due to heart failure or fluid overload. So essentially you rule out other causes of a worsening pulmonary picture, and you wind up with the acute respiratory syndrome. And this is pathologically what you see. This happens to be the results of an autopsy from a patient who died early on with COVID-19. I think most people are aware that there are 3 general risk factors for severe disease. Number 1, and probably the most important, is the presence of comorbidities. This should come as no surprise to anybody, as comorbidities are associated with worse outcomes of many, many diseases. Age is also a risk factor. The older you are, the worse potential you have for, the potential is for a worse outcome with COVID-19. And then male sex is also a risk factor for severe disease. So this then brings up one of the questions that I think really needs to be addressed by research, and that is the association between aging and severe COVID-19. So some of the answer here is trivial. As I mentioned, comorbidities clearly play a role in the age-related association between in the association between age and severe COVID-19. But it's not the only reason, we think, because there are people who don't have comorbidities, but are in the older age group and have severe disease. And by the way, I should point out that the same association between age and severity is seen in SARS and also in MERS. So one other potential reason that older people fare worse is because of waning immunity with aging. But this is not so straightforward, because when we see a poor immunity associated with poor outcomes, we often see that at both extremes of the age spectrum. So we see that in really old people, but we may also see that in really young people. But that's clearly not the case with COVID-19, where we only see severe disease in older individuals. Well, it turns out that this story, this association between age and severity, is probably pretty much a complicated affair. And in fact, it can be modeled in mice. So this is a study where investigators wanted to look at the association between age and SARS in a mouse model. So they used a mouse-adapted mouse-adopted virus, and they gave it to young mice, 4 weeks of age, and also to older mice, 6 months of age. Now, mind you, these are not super old mice, but nonetheless, that age difference resulted in a huge difference in disease expression, with the older mice getting sick and dying, and the younger mice really tolerating this infection quite well. In another study, looking at the same question, that is the association between aging and severity in SARS, again, not COVID-19, but SARS, what they found was that the younger mice upregulated lots of genes, as did the older mice, but the older mice did it sooner, within 24 hours after infection. So there was a delayed upregulation in gene expression among the young mice as compared with the older mice. And then by 72 hours, things were equal between the two age groups. So I think really what this shows you is that this area describing the inflammatory response and how it's associated with age and how it impacts COVID disease expression is really, I think, an area that's very important to investigate. The next interesting question, I think, that really clinicians are particularly interested in, as well as others, is why do men seem to develop more severe disease? Well, one reason is that men tend to have more of these comorbidities that are associated with severity. They tend to be smokers. I guess men just don't take care of themselves as well as women. And perhaps more profoundly and more interestingly, it's very well known for a lot of infections that women do better than men. But the downside of this enhanced immune response to infections in general is that women also tend to get more autoimmune type diseases. And at least some of this is likely due to the effect of estrogen. So here's a study by Stanley Perlman, again with SARS, where they looked at male and female mice. Again, this is a mouse-adapted SARS, and you can see that the males die much more rapidly, more readily than do the females. Interestingly, if they took out the ovaries of the female mice, they behave like men, like male mice, and they died quicker. And similarly, if you gave the female mice a estrogen antagonist, they also behave more like the male mice. Again, I think there's something very interesting to look at, an area that's ripe for research when associating the effects of sex with outcomes in COVID-19. Another particularly interesting area that I'll mention is the role of ACE inhibitors in COVID-19. So ACE inhibitors are a class of drugs that are used very, very commonly to treat hypertension, heart failure, and other entities as well. And as Mike said, ACE2, the angiotensin converting enzyme receptor 2, sorry, the angiotensin converting enzyme itself, number 2, is a receptor for SARS-CoV-2. And it turns out that the renin-angiotensin system is involved in the pathogenesis of the acute respiratory distress syndrome. And ACE2 itself appears to counteract the pathogenic effects of RAS activation on the pathogenesis of ARDS. So that suggests that the more ACE2 you have, the better off you are going to be if you get an infection that has a tendency to develop into ARDS. So these ACE inhibitors that many people are treated with, it turns out they don't bind to the ACE2 enzyme. However, there is evidence that they, through a rather complicated pathway, they increase ACE2 expression in various tissues. So what this means is two possibilities in terms of the potential for ACE inhibitors to affect the outcome of COVID-19. One is that it increases the availability of receptors for the virus. This would not be a good thing for the host. However, again, if you increase ACE2, you might assuage some of the pathogenic effects of RAS activation on the development of ARDS. So we don't know yet. You would think that this would be a question that could be answered very quickly by looking at clinical data. And right now, the only clinical data we have where they compared people who had taken ACE inhibitors with those who hadn't at the time they were infected with SARS-CoV-2 suggests really, if there is an effect, it's very mild. It seems to worsen the disease, but not statistically significantly. And the sample size is really too small to really draw any conclusions. So I think a lot more to come on this issue in the future. Another important question for all of us is will subsequent infection with SARS-CoV-2 be possible? And of course, the corollary to this is, will there ever be a vaccine? Because if you would think that if subsequent infection was possible after COVID, that we'd have a very difficult time developing a vaccine. Well, it turns out that many viral infections provide long-lasting immunity, measles, for example, just to name one, but some don't. And unfortunately, coronaviruses as a group are one of those that don't seem to engender long-lasting immunity after infection. So, for example, you can become infected with the common human coronaviruses repeatedly, though subsequent infections are probably more mild. And in reality, it's probably a situation where you develop an immune response to one of these common coronaviruses, and maybe it protects you for a year or two, and subsequently you're at risk for repeated infections. And this kind of is similar to what we see with SARS. Now, nobody's ever been infected more than once with SARS, as far as I know. But if you look at this graph on the bottom, showing the percent of IgG positive, these SARS-specific antibody positive individuals, after they've had SARS, you can see that it peaks, as do the titers of these antibodies, at about 90 to 120 days. And then it rapidly declines, such that by about 3 years, only about half the people still have anti-SARS antibodies in their serum, and these tend to be of low titer. So it'll be very interesting to see if there's long-lasting immunity after COVID-19. One thing I could tell you is if you infect a monkey with SARS-CoV-2, they mostly recover. If you wait 28 days and reinfect them, they don't get infected. So that's somewhat reassuring that if you respond favorably to a COVID-19 infection, that you won't be infected a month later. But after that, we really can't say.
1 minute warning, Don.
Okay. And this is my last slide, in fact. So the next obvious and important question is there effective treatment for COVID-19? And really the jury is out. People are looking at hydroxychloroquine, they're looking at polymerase inhibitors, and they're looking at protease inhibitors, both protease inhibitors that inhibit the viral protease, but also a particular host of protease, which is important for SARS-CoV-2 replication. And then finally, antibodies may be effective therapy. And I did have one more slide just to mention that 2 animal models that I know about. One is the human transgenic, the mouse that's human, that's transgenic for human ACE2, and then monkeys, but perhaps Ilham could say more about this, but monkeys, while they do get infected, do not seem to develop very severe infection. I will stop there, and thank you.
Okay, so thank you, Don. I'm next. Okay, perfect. Okay, so can everybody see my slides now? So I'm going to
Yes, we can.
yes, perfect. I'm going to talk to you about looking at the immune response to SARS. And so the first thing that I want to bring up is remind everyone that the lungs are actually not an immune privileged site. The lungs actually are very replete with immune cells. And this is just a very simplified graphic of the type of immune cells that we can expect to see in the lung. And it really does cover the entire range of the immune response, starting with intrinsic responses, so the mucociliary escalator, the antimicrobial molecules, and our microbial flora that exist in the lungs. The epithelial cells as well are actually equipped with ways to generate antiviral immune responses. And then, of course, on the other side, we have a number of immune cells, including T cells, natural killer cells, dendritic cells, etc. So, and over our course of
Ilham, just
yes?
let me interrupt you for a second. Can you put your slides on full mode, on screen mode, so we can see it? It's a little small the way it is now.
Okay, let me try this sharing.
Just in your PowerPoint.
How's
Yeah,
that?
that's better.
Is that better?
Thank you very much.
Okay, it's just I have a dual screen, so
Thank you very much.
the wrong screen. Okay, so yes, so the immune system, the lungs are actually have a pretty robust immune system, and as we get older, we actually accumulate a number of these immune cells in our lungs. So the number of T cells and NK cells, natural killer cells, increases dramatically with age. And again, just to orient everyone, we generate pretty robust immune responses to viral infections in the lung. So this is a representative example of an immune response to respiratory syncytial virus. And we can see that, can everybody see my cursor? Yeah. So the virus infects epithelial cells. It generates an innate immune response in the epithelial cells. This innate immune response can protect neighboring cells, and more importantly, this innate immune response leads to the generation of inflammatory mediators that can also help destroy the virus. And then we have the sentinel cells called the dendritic cells that will pick up antigen and take it to the draining lymph node, where they will activate T cells and B cells, which can then migrate back into the lung to respond to viral infections. So what do we know so far about the immune response to SARS-CoV-2? These are all very recent papers that have been published. And in this paper, the authors infected respiratory epithelial cells from humans with RSV, respiratory syncytial virus, influenza A, and SARS-CoV-2. And you can see that all 3 viruses generate a pretty robust transcriptional response, with RSV generating the largest. But there is quite a large amount of overlap between the 2 viruses. And when you hone in and look at the type of genes that are upregulated or generated in response to this viral infection, they all belong to a class of genes that we know as the ISGs, or interferon stimulated genes. And these play incredibly important role in antiviral defenses. And again, just to put this in perspective, this is a heat map looking at gene expression changes with infection in vitro in the human epithelial cell lines. And this bottom row is the respiratory syncytial virus response. This is the response to SARS-CoV-2, and this is the response to influenza virus. And what is immediately apparent is that the number and the magnitude of induction of interferon stimulated genes is largest with RSV. It's also quite robust with SARS-CoV, but not to the same extent as RSV. And what we don't see is this downregulation of genes genes that influenza is actually pretty good at doing. So this set of red genes over here and over here and over here are actually influenza virus downregulating the interferon response. We don't quite see that with SARS-CoV-2. So the interferon response is incredibly important because it generates an antiviral state, not only in the cell that gets infected, but in adjacent cell, thereby protecting the entire environment. However, too much interferon response is actually quite pathogenic. And this is seen here in this representation. So there is a sweet spot for the type of interferon response that you want to generate. So you make a little bit of interferon, that's great, and creates an antiviral state. You make too much interferon and you actually start killing off your T cells and causing a lot of pathology in the lung epithelium. And this is really important because I think the data that we're seeing right now seems to indicate that in the severe cases of COVID-19, we are seeing some of this potentially interferon-mediated immunopathology. So I do have some data from SARS-CoV-2 infection in human lungs that I'm going to show, but this is actually data from the original SARS virus in monkeys. And this is comparing the immune response in young adults and aged macaques. And you can see that in the aged macaques, you have a much more severe pathology score. And you can see that in the lungs. These are the lungs from the aged monkeys. They look pretty devastated. And you can also see the pneumonia in the aged lung that's a lot more severe than the young adults. And what I wanted to point out here is all of this occurs in the absence of any differences in the viral loads. So this is occurring independent of viral load. And subsequent studies by the same group showed that what is happening in the aged lung is that there is an overexuberant innate immune response. And so if you look at the list of genes, and in this case, red means highly upregulated, green means not so expressed, you can see that in the aged lung, you have a lot more clusters of genes that are highly upregulated. And this leads to the immunopathology in the aged lung in this model. But what's also reassuring is that there is a very quick window in which an interferon boost can actually help. So in the study as well, what they did was treat aged macaques during the first 48 hours post-infection with pegylated interferon, which is what we used to use for Hep C. And those macaques actually had a lower viral load and survived much better. So what I was trying to say with all of this is that interferon is important at the right time and in the right amount. Too much of it can actually lead to immunopathology. And so this is kind of a schematic of what happens in an early stage immune response where you have the right induction, but what happens in a severe stage where you have an overexuberant immune response. And in this review article, the authors suggested a number of things that could potentially be used to mitigate the disease severity, including using interferon alpha or anti-sera to boost the immune system. And I guess as a note of hope, in this other in vitro study, this is, again, cells that are infected with SARS-CoV-2 or SARS. And if they are pretreated with type 1 interferon, you can actually cut down the viral replication of SARS-CoV-2 more than you can with SARS virus. So then just to kind of, again, reinforce the message that the right amount of interferon in the right window of time could actually potentially be very helpful here. So there's a number of papers that have been published fast and furious in the last 2 weeks looking at adaptive immune responses in humans infected with SARS-CoV-2. When this paper looked at one individual who had mild disease, and what they were able to document in this particular subject is that there was a very short course of viral shedding seen here in the nasopharyngeal swabs. They did detect viral RNA in the feces and in the sputum, but the decrease in viral loads coincided very nicely with the development of IgG and IgM antibodies, as well as the development of a T-cell response that was helpful to the generation of antibodies. So this little line in black is a subset of T cells that is known as the follicular helper T cells. They're very important for generating antibody responses. You can see that they are dramatically increasing, and they continue to increase long after this patient has actually stopped shedding virus. And there's a peak of antibody secreting B cells that corresponds to the clearance of viral shedding as well. And I cannot emphasize how important it is to actually look not only at viral shedding, but antibody response. So this is a really nice study as well that documented the induction of IgMs within 8 days post-infection or post-presentation to the ER, IgA antibodies, which are important for mucosal defense, and also IgG. And in this study, when they coupled PCR with immunoglobulin measurements by ELISA, they were able to actually more accurately estimate the disease burden in their population. So that's just something that we need to think about in terms of how we do our surveillance studies. Not only is the B cell response important, but so is the T cell important. So again, in that same patient, we have a very nice increase in the number of CD8 T cells and CD4 T cells after infection, and those high numbers persist. Day 20, this is 13 days after the patient has actually stopped shedding virus. These T cells are all equipped with molecules that help them kill viruses, such as granzyme A, granzyme B, and perforin. So they are very effective at killing virus. However, to go back to that balance between innate and adaptive immunity that I mentioned in the slides when I showed the response in monkeys, this is a longitudinal analysis of gene expression profiles in subjects, 3 cases that were infected with SARS-CoV-2, and this was done in the blood. And all I want to point out to you is case 1 was the most severe, case 2 and 3 were mild. And in case 1, what you see is an upregulation of this one cluster of genes that was mostly inflammatory genes that doesn't exist in the mild cases. Whereas in the mild cases, they have an upregulation of this cluster of genes, which is important for antigen presentation. And antigen presentation is what you need to get T cell responses off the ground. So it seems that, like described, as previously described in the monkeys with the original SARS, that an induction of an aberrant inflammatory response right off the bat is not necessarily the greatest thing, whereas an induction of a response that leads to antigen presentation and T cell immunity may be what we want to do. And then these are my last 2 slides. This is a really nice paper with single-cell RNA-seq from bronchial alveolage fluid from subjects who had either mild or severe COVID-19 infection. And they were able to compare that to the bronchial alveolar lavage fluid from healthy subjects. And they had 8 subjects per group, so it's actually a pretty well-powered study. So what I want to point out, so this is our healthy group, and this is a circle that tells you the composition of the different cells in this BAL fluid. And what you see with, in the mild disease, you have an expansion of T cells. This is the blue color over here. You also have an expansion of these myeloid cells, which are usually the source of that aberrant inflammation. And what we see in severe cases is that there is still an increase in the frequency of T cells, but there is a much more significant and dramatic increase in those myeloid cells. And if you move over to these very funny plots, these UMAP files, the only thing that I wanted to highlight to you here is that in the healthy control, you have this nice big cluster of red cells that disappears in the severe cases and is replaced with this cluster of blue and green cells. These red cells are alveolar macrophages that normally reside in the lung, and they do their job by housekeeping and cleaning out pathogens. This group of green and blue types of macrophages are highly inflammatory macrophages that lead to fibrosis. And so you can see that in the severe cases, you have this switching of the types of macrophages that live in the lungs that is consistent with the development of that aberrant inflammation. And then when we look at the T cell subsets in this paper, we kind of see the opposite of what we saw with the macrophage subsets. So in the mild cases, you have this expansion of these pukey green cells. These are actually CD8 T cells. And in the severe case, you have a little bit of an increase, but they're not as dramatic as the mild case. And this is again illustrated in these bar graphs. So that pukey green color you can see is most increased in the mild cases. And not only do CD8 T cells numbers correlate with the outcome here, but in the mild cases, you actually have, if you look over at these donut circles down at the bottom, you have a large percentage of these cells that are in the green, which means they're highly diverse T cells. That's really good. In the severe cases, you have more of what we call clonotype, clonal expansion. So very few T cells are actually expanding quite dramatically. And diversity is very important when it comes to T cell immunity. And so I will end with just saying that while there is a number of really nice studies that are coming out right now, we still do not fully understand the relationship between host immune responses in the lung and the periphery, and then how those correlate with disease outcomes. We don't fully understand the mechanisms underlying increased susceptibility in some individuals. As Don said, it's very complicated. We also don't really appreciate the role of cellular, so that's T-cell mediated versus humoral, B-cell mediated responses and protection. We need to find ways of regulating aberrant innate immune responses, and we don't know how. We also don't know what protective immunity looks like and what its longevity is going to be. And then in terms of vaccine development, we don't know what should be included in a vaccine and how that's going to be helpful. And there's lots of questions revolving around. So I just looked at the CDC's report on morbidity and mortality related to SARS-CoV-2 that just came out. And this looks like the statistics is out of the known cases, 7% of them were hospitalized and 1.4 ended up in the intensive care unit. But there is this large question of asymptomatic individuals. So those 91% of the individuals who had mild disease that did not require hospitalization, do they shed as much virus as symptomatic individuals? Are they really these large carriers and super infectors out there? We still don't have any of those answers. So I think that's a really good segue for Andrew, Dr. Neumer, to then talk about surveillance and how do we calculate disease prevalence, etc. So, Andrew, I think you should be able to now, I'm going to stop sharing so that you can share your screen.
Okay, hold on one second. I am sharing. Okay, can everyone see that? Or
I can see it, Doctor.
Great. Thank you. Thank you all for coming. I'm Andrew Neumer, Public Health and Center for Virus Research. And I'm a population scientist, so I don't work with the lab in a clinical setting or in the laboratory. But I've been highly involved in the COVID-19 on several dimensions. My principal area of research is mortality, human mortality at the population level. So not predicting, which hospital or clinical patients will have the best outcomes, but just studying patterns and processes of mortality at the national and global level. And mortality is a lagging, not a leading indicator of a pandemic. So, and to make matters worse for my studies, the mortality data are compiled with an even longer lag. So what I've been spending a lot of time doing in the early days of this pandemic is working on public outreach and trying to get people to understand what's going on. So I've been doing a lot of that work. As you all know, we're in the so-called lockdown right now. I think one of the things that's going to be super important as far as the timing of when that ends is to start doing serosurvey panels with large sample size that are scientific samples, nationally representative, and tracking, when I say panel, I mean tracking the same people over and over again and redoing serology so that we know what the population level immunity is. And that sort of dovetails with Ilham's last remarks about, the immunity, and also some of Don Forthal's remarks about the persistence of the immunity and the prevalence of asymptomatic infection. And the CDC should use the NHANES, the National Health and Nutrition Examination Survey. They should use the NHANES sample frame and start doing serology studies now. I've also been working with the OCHCA under the leadership of Dean Bernadette Bowden-Albala of the Program in Public Health here at UCI to come up with some estimates of a surge in Orange County hospitals. We're seeing more and more cases here. We're seeing cases county-wide, and hopefully we can stay one step ahead of the virus. So how long have I been warning about the virus? Well, on the 31st of January, after a long series of messages warning this was coming and some arguments with people that it was going to be worse than flu, I said, duct tape your underpants. 2020 is going to be a wild ride, hashtag coronavirus. So I've been raising the alarm about this for some time now. So let me say a few words about how we should think about the mortality effects, because this is my most specialized area of expertise. And then I'll probably run slightly less than my 10-minute allotment, so we can have time for questions, if that's all right with everybody. Questions to the whole panel of presenters, of course. So
Andrew, this is Bert. Don't rush this. Go ahead and go.
Okay. Well, I should be able to fit this all in just under 10 minutes. But so COVID-19 mortality is obviously something, it's the most extreme outcome. And it's something that therefore everyone's been talking about. And of course, nothing captivates the public imagination quite like mortality, right? And so there are really four principal ways we need to think about COVID-19 mortality. And then there's even more ways in this fifth category. But I call these direct, indirect, and competing risks. And so let me give you a thumbnail of what these are. So the first category is direct. And so basically these are the ones that we've already been hearing about, okay? These are deaths that are being counted as COVID mortality in real time. So they're showing up in the newspaper website. They're showing up on the Johns Hopkins dashboard. There's a few others of so-called dashboards on your various websites and what have you. That are tracking coronavirus mortality. And these are, they're going to be assigned ICD code U07.1. It's going to be pneumonia as the proximate cause of mortality, and COVID or coronavirus as the underlying cause. And these are being counted, and these are the deaths that we hear about, every day, every evening. And this is what everyone's talking about when they say, oh, there's been over 2,000 deaths in the United States right now. But we actually know that this is going to be an undercount. These direct counts are going to be an undercount of coronavirus mortality. And it's going to be quite some time before we have a complete reckoning of all the mortality impact of COVID-19. Just yesterday, there was an Italian newspaper that published data for one city, which showed that COVID-19 deaths had peaked in this city, but that all-cause mortality had really spiked, like 5 or 6 times higher than the number of coronavirus deaths when compared to an average of daily mortality over the previous 5 years. And it's clear that some of those all-cause mortality increase, which are not being counted as COVID, have something to do with what's going on right now. So we're going to have to really go over the data carefully. The direct indirect effects are not a direct tally. They do not appear in the dashboards we've all been following. These are early COVID deaths that were mistaken for influenza, and later COVID deaths that will somehow not be recorded as COVID. So this is a pneumonia death where coronavirus is not mentioned on the death certificate. So these will be coded as ICD-10, J11, or J18 typically. And these are not showing up in the dashboards, as that Italian example illustrates. So we are already undercounting COVID mortality, because not every death certificate will say coronavirus, and therefore it will not be counted as ICD-10 U07.1. It will be counted as ICD-10 J18. Different places have different customs about how sure they are about something before they can write it on the death certificate. The third type is indirect, and this is really a massive category of mortality that we haven't seen in the United States so far, I think, for the most part, and that we're really hoping to avoid. This is why the U.S. naval ship Comfort is at anchor in New York Harbor, 1,000 beds on a Navy hospital ship to treat non-coronavirus morbidity. Why? Because they want to be a pressure release for New York City hospitals. They are not treating coronavirus cases on the USNS Comfort. If you have a broken femur, which as you know is ICD-10 S72, that can be fatal, but typically it's not, because emergency departments are very experienced at dealing with broken femurs from automobile accidents. But if the emergency department is completely inundated with COVID patients, then a broken femur can be fatal. And this is exactly the kind of thing we're hoping to avoid by flattening the curve, by social distancing, so hospitals can keep operating with some semblance of normalcy. And the next category is competing risks. And this is something demographers think about all the time, and non-demographers sometimes do and sometimes don't think about. And competing risks isn't a category of deaths per se, but it's highly relevant to our conversation today. And that refers to the fact, and particularly since most of the COVID-19 mortality has been among older people, that someone who dies of COVID-19 now in March or April may have otherwise been quote unquote destined to die, let's say of a heart attack during a July heat wave or what have you. So you can only die once, and therefore the heart attacks mortality in this hypothetical July or August heat wave will be lower than it otherwise would be. Now, when you think about you can only die once, there's always this sort of thought experiment that demographers do, that you're saved from, if saved or killed by the first cause of death, and therefore killed or saved by the second cause of death. And the point is that these come into play in some cases in relatively large magnitude. So we are going to have to pore over the mortality data for the whole year in quite some detail before we fully understand the mortality impacts that this is having. And the NCHS releases mortality data typically with a 2-year lag. I'm sure they're going to do everything they can to expedite the calendar year 2020 data. But nonetheless, you know, it's going to be a while before we know all the contours of COVID mortality. And, you know, it's important to remember that these dashboards are not counting all of these other nuances. They're only counting deaths that are coded as COVID deaths. So the dashboards are counting a simple tally. They add up every day more and more deaths, but these effects are not strictly speaking tally deaths. These are interaction effects. So it's going to be a long time before we understand COVID mortality in all of its nuances, but we should remember that we're probably underestimating it now, and that we should bear that in mind as we think about, you know, the impact this pandemic is having. People have also suggested that the coming recession will bring economic and psychological effects, which then act back on mortality. That's a little bit further afield, but it's something that could be real. And then people have postulated that there may be scarring among survivors, so that survivors of severe infection may have enhanced mortality for some years to come. And, you know, that's something that we can study, hopefully with some panel data, but it's nothing that can be dismissed out of hand, although some hypotheses about the 1918 flu along the same lines haven't really stood up to scrutiny as far as we can tell. So it's just going to have to be a wait and see approach. And so, like I said, we're going to have to pour over the final data, mortality data for 2020, and do a lot of indirect estimation. This is sort of my specialty, so I'll certainly be involved in all of this. And for a point of reference, the 2018 final mortality data were released about 6 or 8 weeks ago. So there's just a lag. There's about 2.8 or 2.9 million deaths every year in the United States. And so it's just a lot of data for the NCHS to go over and process every year. So that's why there's a delay. They get the data from the 50 states and they act as a clearinghouse. It takes time. There could be anywhere from 50,000 to 1 million coronavirus deaths in the United States in 2020. I've been saying there could be up to 1 million deaths since January. I'm on the record of that. And I still think there could be up to 1 million deaths. But it could be as low as 50,000. It's really too early to tell. So that's all I have to add. Thank you very much.
Thank you, Andrew.
Trying to
I know
unshare.
that some members of the panel actually have to go to another meeting in a little bit, but I think we have time to take to answer a few questions. So I've been scouring the chats and I've tried to group the questions into themes and then direct them to the appropriate person. And so one of the questions I think, Dawn, you'd be great at answering is, does flu cause ARDS as well, or does it not? So the question was, you know, does flu not cause the same type of opacities that are seen with SARS-CoV-2?
Yes, it can. In fact, just about any virus that attacks the lungs can cause ARDS, as can many other things. So the answer to that one is yes. Fortunately, or unfortunately, it doesn't do it as often as any of the SARS virus, or any of the severe SARS viruses.
Another, I thought, really relevant question along that same vein, so probably Mike or Don, any insight into why the original SARS outbreak actually was more easily contained than the current SARS outbreak? So I think we all have a lot of thoughts about that, but maybe I'll take, Mike, you want to take a crack at that? And then Andrew looks like he has something to say too.
Yeah, we like to think that it was contained because it was a less virulent virus, but we really don't know that. If you recall, the 2003 epidemic was accompanied by an enormous amount of publicity. There were people monitoring airports. I traveled to China twice during that year, and each time when you came off the airplane, you know, you were monitored in terms of body temperature by a remote infrared monitor, and if your temperature was a degree above normal, you were sent into another room for a complete physical to ensure whether you were infected or not. So they had pretty tight control over things, but I think also, you know, the 17 years that's elapsed has really brought a much higher standard of living and mobility to people in China as well. You've got people with the resources to travel. This happened to start during the Chinese New Year. So you had people traveling extensively within the country, and even people from far afield like the US, you know, going back to China for Chinese New Year. So there were a lot of factors interacting. I don't think that we know whether or not this virus really is inherently more dangerous or not. I think it probably is a good thing to understand.
Hi, Ilham. Can you hear me?
Yes,
Yeah.
we can hear you, Bashir.
Yes. Hi. This is Ben Mohammed. I have two questions. One is for those of us who are developing vaccine for coronavirus. I hope that UCI officials are listening. What can we do today to speed up an IRB and an ICOC, common probably for all of us, so we can get access to patients when they are here? The second question I have is, I noticed, Ilham, some of your slides, it looks like CD8 T cells are more associated with asymptomatic infection than do CD4 T cells. Do you see any other studies where they look at for CD8 function, exhaustion, and all this, and correlate that with symptomatic versus asymptomatic?
Okay, so I can't spot. I know that the IBC, IACUC, and IRB are actually fast tracking everything that has anything to do with SARS-CoV-2. So if you put in an application, as the chair of the IBC, I can tell you that we are working around the clock to review those as they come in. But to go back to the CD8 T cell response, so there's only one paper that looked in the lung, and that's actually one of the things that I wanted to say is, so there's a group of us who are trying to set up a biorepository of samples here to look at and be able to study. And that is the only study that has looked at the lungs. And that's missing because it's really hard to sample the lungs and look at the immune response in the lungs. And so it's only one study. And what they were able to show is that there is a higher frequency of CD8 T cells in the mild cases compared to the severe cases. And so at this point, we don't know if that's just an association, if that's the canary in the coal mine, if that's actually functionally different than what's happening in the severe cases. So I think we just need to do more work. I think that's what it comes down to.
Thank you.
Ilham, if I could address the question about why SARS may have died out.
Yeah, I want to go back to that. And then I think Andrew really wants to say something too. So Don, yes.
Okay, yeah, no, I have a very simple answer to that. I don't know if it's true, but from my understanding, the ability to isolate SARS-CoV virus out of the upper airway is much less than the ability to isolate SARS-CoV-2 from the upper airway. So it may simply be that there's just not a lot more coughed around. Now I know there are these super spreaders of SARS, so they may be a special case, but in general, it looks like it was just more difficult to get the virus out of the upper airway.
Andrew, do you want to
Yeah,
add
well,
something from
yeah,
the
no.
epidemiology standpoint?
Well, I endorse what Dr. Forthall and Dr. Buckmeyer have said, but I also think of two other things. First of all, I think we may just have caught in a break and jumped on it early, and just if you rerun the universe starting in 2002, then there could have been another outcome, and just sort of one of those random things. But also, SARS. It seems like it's more likely to land you in the hospital, so it's more likely to get you isolated, and there's probably fewer asymptomatic spreaders. I mean, there's still a lot we don't understand, as you and others have mentioned about asymptomatic spreaders in the current pandemic, but none of the epidemiology we've seen is compatible with zero asymptomatic spread. So there's at least some. And we don't know to the extent to which that was the case with SARS.
There was a couple of really good questions from Peter, actually, about reservoirs for SARS and animal models. And so, as Dr. Buckmeyer, I think you could take this on. So we know this is a zoonotic disease, which means it originated in an animal reservoir. And so the question is, can other animals in the United States, like ferrets, domestic cats, could they potentially be acting as now a new reservoir for this virus here? And the counterpart of that is, will they be potentially good animal models to study this? Mike?
I'm sorry, I didn't realize I'm still muted. Oh, I guess I'm not. Okay. I didn't realize you were calling on me. The problem with SARS, again, was that we were operating in a different era. We had much better leadership from federal government in the States. We had very, very few incidents of even suspected SARS in the US, and even fewer cases. And you didn't have the distribution to faraway places, in mass, like India and the African countries. And, there were very few cases that escaped Canada and Europe and China. So it's just very hard to say what would happen if the SARS virus hit again. I don't know. It's inherently a higher mortality if you believe the mortality figures.
Right. So Mike, what do you think about domestic cats and ferrets and other animals now acting as reservoirs for this virus in the United States?
We heard all that before, and it turned out to be nonsense. If a domestic cat has SARS, who is it going to spread it to? The immediate family. How is it going to get SARS? Certainly not in University Hills, where everybody's cats are indoors. And I don't think coyotes are intermediate animals. I think people spread that kind of stuff like they tell you to gargle with salt water because the virus can't stand salt water. Things like that pop up in the popular press and on the list servers and places like that that really are just speculation or absolute falsehoods. So I think that if there is an infection in cat, probably the cat dies. Civet cats don't die, but civet cats are not felines. Civet cats are a very different animal. So I don't accept that. The number of cats that could be infected would be a very, very small fraction of the number of animals that you'd need to sustain an epidemic. There was a rumor that in the Amboy Towers in Hong Kong that there was spread via rats, but that turned out not to be true. It turned out that it was the ventilation, cross-ventilation between the bathrooms and the kitchens in the ventilation towers that probably spread the virus, that aerosol was going directly from one to the other.
Okay, so unlikely to potentially get a new reservoir here.
Yeah, I think so.
Okay, I think there's also questions about what is known about the fidelity of viral replication and how many mutation, what's the rate of mutation, or how mutagenic is this virus?
How mutagenic is it?
How likely is it to acquire a ton of mutations? And as a follow-up question, is that likely to attenuate or make the virus more virulent?
No, I think with every round of replication, you get a selection of mutations that occurs, and those are chosen really at random, and they only have an effect if they affect replication. Now, so you can have what would be a silent mutation for replication in tissue culture, and it may have a very profound effect in vivo. For example, by passing in neuronal cells, we were able to select a virus that fundamentally changed its route of entry into the cell, from a neutral pH fusion at the plasma membrane to an endosomal entry requiring an acidic phase to get into the cell. And we did that over 60 days by passing in olfactory bulb neurons. And we know exactly what the changes were. And it turns out there were only about 9 changes in the genome, in the entire 31 kb.
Yeah, no, I think that those viruses, it also mutate to have better entry too, right? The mutation. And when you look back at the Ebola, so the big Ebola epidemic that occurred between 2014 and 2016, if you compare the host response to the later isolates of that virus compared to the early isolates, actually the host immune response became more adaptive. The virus and the host immune response became actually less adversarial than it was at the beginning of the infection. And so it's just anybody's guess whether it's going to go one way or the other.
A fundamental rule of viral biology. The virus that kills its host is unsuccessful. The virus that survives in its host and is excreted for a longer period of time is more successful. Mm-hmm. And so anything that would reduce the mutation, would reduce the pathogenesis of the virus, but at the same time allow the virus to replicate and be excreted, then yes, you'd expect that. And we do see that in a number of infections. The virus that we passed for 60 days was less virulent than the one that we didn't pass by really over a factor of over 1,000 in LD50.
Also, another question that I saw, is there something special about the bats in China? Or can bats everywhere be carriers of coronaviruses?
Bats everywhere carry viruses.
Coronaviruses, we're talking about this.
Species. It happens that the horseshoe bat is one that is frequently sampled and targeted in China, and it doesn't carry just the SARS-like viruses, it carries other pathogens as well. But I think it just depends on where you're looking. Don might know that better because Don, was in Africa and was very interested in Lassa fever and rodent-borne diseases, and may well comment on that. There are going to be different rodents and different bats, no matter where you look. After the SARS epidemic, Catherine Holmes organized a group of people to sample bats in Colorado, and they found SARS-like viruses in Colorado. Now, how close to SARS, we don't know. I don't know that they ever published it.
There was also a question about animal models, and I don't know, Don, if you want to expand on that, but rodents do not express the appropriate receptor, but there are transgenic mice, right? Don, you want to say something about that?
Yeah, that's where rats and mice don't seem to have the ACE2 that will bind to the spike protein of SARS-CoV-2. So what has been used, and there is a publication on this, is a mouse that is transgenic for the human ACE2. And it seems to recapitulate some of the findings. These animals do get sick and you can isolate a virus from them. So it seems to be at least a reasonable model.
Can I add, Ilham, if you don't mind? We do have the transgenic mouse that express the human receptor. So if you guys need it, we can share it with you. We ordered it. It should be here in few weeks.
I will take you up on that, Basha. Thank you.
And Ralph Barrick told me that he had to adapt the virus a little bit to actually get good infection in those transgenic mice. So that's just an FYI. Okay, so there's a couple of questions that have been asked about what kind of systems might be set up to distribute samples. And all I can say right now is that the ICTS, the CCR, and a lot of us are working really, really hard to establish an umbrella IRB, a distribution system, a sample procurement, consent forms. As you can imagine, these are very challenging circumstances to actually get consent, get the right specimens. The pathology labs are slammed. And so we are working really hard on figuring out the best and the most sensitive way to both the patients as well as the healthcare providers to obtain and cryopreserve cells, freeze samples, and then establish a repository that will be then available to people where they could put in an application to receive samples, and those applications would be reviewed by a committee of scientists, and the process will be very transparent, and we are trying to accommodate all those competing research needs. Okay, I'm trying to scour the chat messages to see if, okay, yeah, so there's a question about whether vaccine studies will be very straightforward as, will vaccine studies be very straightforward, and what are the likelihood of generating a successful vaccine? I don't know who wants to take that on.
Yes, so we started, this is Ben Mohammed. We started mapping all the epitopes, CD4 epitopes, CD8 epitopes, and Tcell epitopes using the algorithms that are available. And we have identified all those epitopes from the whole genome, from the 12 proteins. Again, if somebody wants to peptides, we can share small amount of those peptides with you guys. So there is an ongoing vaccine development that is going to use the transgenic mouse that express the human receptor, that we are actually crossing with HLI transgenic mice that is expressing human leukocyte class 1 and class 2. So we have a mouse model that both express the receptor of the virus, at the same time it expresses human leukocyte antigen, which is going to be a very good animal model, humanized model to use. So we can share that, we can share the epitopes. What I want to know if we can get together and then probably look where we can submit a grant where we can have everybody's expertise, and it's going to be the grant for COVID vaccine at UCI. So I want you guys to think about that.
So I know that several members of the panel actually have to run to a 3 o'clock meeting, right, Bert?
That's correct.
Okay, so I think we will stop now, but we do have, all of this will be recorded, it will be available on the YouTube, the chats will be available, and maybe there will be a follow-up process to answer some of these questions and create some of these questions.
Thank you.
Bert, do you want to say any closing remarks?
Yeah, I'd like to thank all of our speakers today for their very informative, insightful presentations in helping to provide a roadmap for the way forward in fighting this pandemic caused by SARS-CoV-2, and all of you online attendees for your valuable participation in this very important communication. Be safe out there. Goodbye.
Thank
Thank you, everybody.